lecture. So we move on to another segment, which is enzymes and digestion. Check out that animation. That's an enzyme with the substrate binding to it, then the product is released. So initially what we had was sucrose, which is a disaccharide. It means it has two sugar molecules. Then when that reaction has taken place, see what happens. Glucose and fructose are the substrate formed. So this is the active site. Can you see it binds into it? They adjust itself to one another. Then the water molecule binds to that. And that's like hydrolysis. When water is added to it, that's hydrolysis. Then it breaks down into that. So let's see what we're trying to do here. We've talked about how enzymes work. Really, we want to see the effect of, how enzymes will work basically in digestion, really in digestion. So let's work this together. Now, the purpose of digestion is to break down substances from their large, complex, insoluble form into what the body can work with. Because the point is, all the whole thing we eat, except for the fluidy ones, they have to be broken down into what can be absorbed in the bloodstream, then from bloodstream into the cells. So it says here that large substances like protein, starch are made from smaller chains of molecules which are held together by bonds. So those bonds need to be broken so that the smaller components can be released. So what enzymes do is to help to break such molecules into smaller ones. For example, what you saw in the previous slide here is just a very minute one. Starch, for example, has hundreds of something like this. So you have to break the bonds. This bond, has to be broken. That's what we're trying to achieve. Can you see that's where it is? What is bounded is broken, then it's released, so to say. So any of such a reaction is called chemical digestion, when enzymes helps to break the food, like we saw in that previous slide. That's called chemical digestion. Now, why do you say chemical? Because we can also break down food physically. That's mechanical, like chewing, but that still has its limits. So, but when it is chemical that is doing it, when it is enzyme that is doing it, we call it chemical digestion. So let's move on. So basically, for the context of this class, we have three categories of enzymes that we use for digestion in humans. We have carbohydrates, which means they work on carbohydrate. We have proteases, which means they work on what protein. They have what lipase, which means they work on lipids, or we say fat, so to say. So these three categories of enzymes are very, very important for anyone to know. Really, don't forget, you can tell what they work on from their names. Really, that's like the truth. So moving on. So the first one on the list is carbohydrates. Now, carbohydrates, basically, like it says, they break down carbohydrate into simple sugar. They break down carbohydrate into simple sugar. Now, you might want to remember what are carbohydrates. Basically, we have rice, beans, all those stuffs. We have juice, but they are not in the same category. For example, starch is a polysaccharide. It means it has a lot of sugar in it. But juice, for example, is sugar. It just has few molecules of sugar in it. So to say, it doesn't, when I mean few molecules, I mean that the sugar is not in a complex form. Yes, basically. Now let's look at this. There's a category of carbohydrates called amylase. It's a type of carbohydrate which is known to break down starch into maltose, which is then broken down into glucose by enzyme called maltase. Now hear this. So any enzyme that can break down starch is called amylase or amylase. Then this amylase breaks down starch into disaccharide. Disaccharide means just two sugar molecules, like maltose that was mentioned here, like sucrose, then like lactose. Now, each of these enzymes, then each of these substrates, disaccharide, it means they have two sugar molecules in them. Now, for example, maltose has glucose plus glucose. Sucrose has glucose plus fructose. Lactose has glucose plus galactose. So each of these are simple sugar, like you don't need to break them down anymore. That's the form in which it can be absorbed. But the body cannot usually absorb these ones the way they are. So can you see how nice it is? So we have starch broken in down to any of these three. Then these three is then broken down into their real form, so to say, into what the body can work with. So let me explain that to you right now. So now maltose can be broken down by its own enzyme called maltase into what? Glucose and glucose. Sucrose can be broken down by sucrase into what? Glucose and fructose. Then lactose can be broken down by what? Lactase, that's the enzyme, into what? Glucose and galactose. This is very easy. So the point is, that's what we say is chemical digestion. The enzymes are the one doing this job. So don't forget, anytime you hear the word amylase or amylase, it means the enzyme work on starch, please. It's not a general way to say all enzymes, sincerely, for all carbohydrates. The general name for carbohydrates, digestion enzyme, is carbohydrates. But the one when we say amylase, we are referring to the one that breaks down starch majorly into maltose. So amylase are made where are they found? They are found in salivary gland, in the saliva in the mouth. Then also in pancreas, we call that salivary amylase. We call this pancreatic amylase, so to say, just the name where they are found. Then there's a name for that pancreatic salivary amylase. We call it thialine. It has its name called thialine, so to say. All right. So in summary, carbohydrate and digestive enzymes are called carbohydrates. The one that works on starch specifically is called amylase. It will break starch into maltose. Then another enzyme called maltase. We now break down maltose into 2 glucose molecules. You can just rewind the video from what I just showed you, not quite long. Then the next type of enzyme is proteases. Proteases. So these are a group of enzymes from the name that works on protein. What do they break protein down into? Into amino acids, so to say. Now, but let me say, they didn't just break down protein into amino acids straight away. It goes through stages from protein to polypeptides to proteases and peptones and the likes into peptides, like that, so to say. I just give a rough overview, but it doesn't just go from protein to, no, it's broken down. I'm going to show you a slide that's going to show you that in a minute. Then the point is, where are these proteases found? They are found in the stomach. The one in the stomach is called pepsin. The one in the small intestine, we have erythrin, we have trypsin. That one comes from the pancreas, so to say. We have other ones, so to say. We have the ones that we have some called peptidases. The whole long and short of the story is proteases are enzymes that break down proteins into amino acids. That's the end product, amino acids. So you can already, oh wow, I didn't even say that here. It says the one in the stomach is called pepsin, and some are in pancreas. That's the one called trypsin. The one from the wall of the small intestine, erythrin, peptidases, and the like, so to say. So this is what I was trying to tell you. Pepsin, trypsin, peptidase. So this is the real protein, very long chain. Then it's broken down into a smaller one, and trypsin will break it down into smaller ones. Of course, then peptidase will just break the final bonds to free each of the amino acids, like that. Hope that made sense. Then the last guy is the lipase. Lipase break down what? Fats or lipids into glycerol and fatty acids. It's very important to remember these things, because you need to know that this is the form in which fat is being absorbed. It can't be absorbed as lipids, never. Protein cannot be absorbed as protein. It will only be absorbed as what? Amino acids. Carbohydrates cannot be absorbed the way they are. They have to be broken down and be absorbed as whether glucose or fructose or galactose, as the case may be. Now, you can see what is happening here. So this is the bond, this is the lipase. It breaks down glycerol off into their fatty acids. These are fatty acids. These are fatty acids. So glycerol is freed, fatty acid is also freed, so it can then be absorbed. So it says they are produced where? In the pancreas. We have what we call pancreatic lipase. Then we have some in the duodenum, as the case may be. So, also they are secreted into the duodenum, because I am not sure the wall of the small intestine does not produce lipase, but it's produced in the pancreas and released into the duodenum. I'm going to show you some of these things as we move on. Okay, I think I talked about this when we did, just check my video on alimentary canal. All right, hope that made sense. Let's do a quick quiz on this. It says, what name is given to an enzyme which catalyzes the breakdown of fats? I guess you say lipase. What is the product formed when fat is broken down? Fatty acid and glycerol, or glycerol and fatty acids. Then suggest what happens. So what part of the body produces this enzyme? Oh, okay, so I think this question is consistent. So lipase. What is formed? Fatty acid and glycerol. Whether you write glycerol and fatty acid, either way, suggest what part of the body produces enzyme. Okay, the pancreas. That's what we call it, pancreatic lipase. Suggest why it is necessary to break down fats in digestive system, so that it can be absorbed into the bloodstream. In the way it is, it cannot be absorbed, so it has to be broken down so it can be absorbed into the bloodstream. Number two, so that cells can be able to use it to form, or the body can use it to form cell membrane.